In 1436A, the question asks us to use the ribosome or translation machinery that we drew in 1435, but I think we should just start over. So let me sketch the small subunit here. That is terrible. Try again. Not so great, but good enough. Okay, so small subunit, and I am just going to diagram the prokaryotic ribosome components. A small subunit here, which is 30S, and this is made of the 16S, there's Svedberg ribosomal RNA, and 21 proteins, or we said over 20 proteins. And we don't normally talk about the individual proteins. There's more than 20 of them. It's way too many to talk about in our introductory genetics class. So, and then we have the large subunit here. Okay, looking something like that. Oh, okay. Large subunit. Large subunit, this is going to be 50S with the 23S ribosomal RNA and the 5S ribosomal RNA and over 30 proteins, right? 31 to be exact. Okay, and if this was a eukaryotic ribosome, we would have another one in here, right? We would have a 5.8 in here. So with this question right here, we are talking about a possible new antibiotic called everneomycin. Okay. And so everneomycin. So this antibiotic here has activity against a broad range of gram-positive pathogenic bacteria. To determine the mode of action, so normally they want to, when they're coming up with these antibiotics, they want to know how they work, right? So they're trying to figure out how does it inhibit translation. So to determine the mode of action, researchers have analyzed 23S ribosomal DNA mutants that showed reduced sensitivity to everneomycin. Okay, so they're looking at a bunch of different strains of bacteria, some that are known to have mutations in the 23S ribosomal DNA or RNA. In the 23S ribosomal RNA. So, and slight mutations in here can kind of make the bacteria slightly less sensitive to the antibiotic. So they seem to have discovered two classes of mutants that confer resistance. So in one class, the mutation occurs in the 23S ribosomal RNA between nucleotides 2475 and 2483. So 23S ribosomal RNA between 2475 and 2483. So these are the positions. So I guess this is a long ribosomal RNA molecule over 2400 over 2483 nucleotides long. And okay, so mutations that occur between these positions in the ribosomal RNA make the ribosome less sensitive to this antibiotic. And okay, so another one, if you have a mutation in L16, which is the name of a protein that makes up one of these, one of these proteins over here, then that also leads to reduced sensitivity to this antibiotic. Okay, next the question says, it turns out that the tRNA anticodon stem loop appears to bind the A site of the ribosome at ribosomal RNA bases 2465 to 2485. So let me see, 2465 to 2485. Okay, that's right in here. All right. Okay, so apparently the tRNA anticodon stem loop appears to bind to the A site of the ribosome at these positions. So I guess the A site, which is here, is partly made up of positions 2465 to 2485 of this 23S ribosomal RNA. And apparently that's where the tRNA, the stem loop of the tRNA, binds when it's being loaded in there for translation. And this also confirms to the proposed function for L16, which appears to be involved in attracting the amino acyl stem of the tRNA to the ribosome at its A site. Okay, so sort of the same thing here. So L16 involved in the same thing. So using our sketch, along with the information I just read, designate where the proposed antibacterial action of evernemicin is likely to occur. Okay, so what have we learned, or what did they tell us? Well, they said that scientists, when they're trying to discover how evernemicin works, they found mutants in here, in this region of the 23S ribosomal RNA. They found bacteria that had mutations in the ribosomal RNA in between these positions were less sensitive to the antibiotic. Same thing for L16. So they found bacteria that were mutated in L16, they had a different sequence for L16, and they were also less sensitive to the antibiotic. So one thing you could keep in mind, should keep in mind, is that these bacteria are still living. So they're still growing, they're still translating, they just have slightly different sequences for the ribosomal RNA at these positions, between those positions, and for L16. So slightly different sequences, but the bacteria are fine. But these slightly different sequences appear to make these bacteria less susceptible, less sensitive to the antibiotic. So less sensitive is more resistant. So it also turns out that scientists already knew that these positions, similar positions, were involved with getting that tRNA into the A site here. So with that information, one, we know two classes of mutations that result in less sensitivity to the antibiotic. So mutations, two groups, mutations equals less sensitivity, and those mutations of the 23S and the L16 are involved with tRNA binding to A site. So I guess all this data, everything is pointing to the likelihood that evernemicin, its mode of action is to bind to the A site and prevent entry of that charged tRNA, right? I think that makes sense. And why does that make sense? So if we were to mutate either the ribosomal RNA or L16 in here, near this A site here, so evernemicin couldn't bind, well then the tRNAs could probably come in. As long as we didn't mutate them in a way that prevented the tRNA from coming in with the charged tRNA coming in with the amino acid. Okay, yeah, so I think so we figured out the mode of action of evernemicin, or have a very good hypothesis on the mode of action.